In last class, we have studied about aseptate of endotrophic mycorrhiza. Now we will continue with the topic, the fungi. Here, only three genera, Indogone, Rhizophagus, and Pythium, are known to form Va mycorrhiza. Indogone being the most important. Only the three genera of the mycorrhiza is there. First one is Indogone, second is Rhizophagus, and the Pythium. Indogone has little or no host specificity. Mycorrhizal species of Indogone are ubiquitous, means seeming to be everywhere, means that Indogone has little or no host specificity, means it is capable of living alone, and it is ubiquitous, means it can be found everywhere. They never appear in list of soil fungi because they are obligately symbiotic and do not come in culture, means that the normally we can find many fungi in the soil, but this is not counted in the list of soil fungi, and it is obligately symbiotic, means both species need it to survive. Both the fungi and the host plant both need each other to survive. In laboratory, when spores are germinated, the growth of the germ tube continues till it is attached to the spores. When the spores is germinated in the lab, it is the growth is continued till the germ spores did not get attached to the spores, means that the germ tube is grown till the time when it does not attach to the spores. Excised, having been cut out, hyphal tip fail to grow. If the hyphal tip is it is when it is cut out and try to grow, it is failed to grow. On other hand, the fungus grow vigorously, means that with strength and with the energy around host root. Species of Indogone are identified by their characteristic spores and structure formed on the internal hyphae. The Indogone species is identified with the characteristics and the structure which is formed on the external hyphae. 5 types of spores and sporocarps are formed which can initiate mycorrhizal association. 5 types of spores and sporocarps formation takes place from which we can identify that it is Indogone species. These spores are: 1. Spores with a bulbous basal portion, also called zygospores. Second one is echinucleate spores formed in holes on a helical hypha. Third is chlamydospores with funnel-shaped attachment held inside a weft of hyphae called sporocarp. And fourth and fifth is spores with simple attachments. The first one is the spores is formed with the bulbous base, and it is also known as zygospores. Second one is the echinucleate spores formed in hole. This one is forming in hole on a helical hypha, the helical shaped type of hypha. It is coiled hypha. This one is coiled hypha, and it is actinonucleate spores. The third one is chlamydospores. These all are chlamydospores with the funnel shape. This is a funnel shape type structure attached with the held inside the weft. This is the weft held inside the weft. it is the chlamydospores, and this is the funnel like base. And the fourth and fifth one was spores with simple attachment, simply attached spores with simply attached hypha, spores with simple attachment, and these are oil globules. So these were the 5 structures of the Indogone species with the sporocarps. Now, the sporocarp was once considered to be the diagnostic feature of Indogone, but now many species are known which don't form the sporocarp at all. The genus includes non-sporocarpic species as well, means that before it was formed with the sporocarp. Sporocarp was one of the specific characteristic from which we can identify that it is a Indogone species. But now many species are with non-sporocarpic species as well. The species are segregated in 2 groups. Now these species are segregated in 2 groups: the chlamydosporic species and zygophoric species. These further divided on the basis of their production inside or outside the sporocarp. Now these are further divided on the basis of the uh, production of the sporocarp, the production of inside and outside, how the structure is forming, how the structure of the sporocarp is inside and how it is the outside of endocarpic and ectocarpic. Majority of species are chlamydosporic. Among these, the ones that form ectocarpic chlamydospores outnumber those that form them inside sporocarps. Now, majority of the species of the Indogone is chlamydosporic. Now, this is the structure. This is the structure showing sporocarp of Indogone species. It may contain chlamydospores or zygospores, chlamydosporic or zygosporic sporocarp. Here it is, we can see that here it is zygospores or chlamydospores, and here it is sporocarp. It may convert into zygospore or may convert into chlamydospores.